Okay, so today is the 21st May 2020, and it's my pleasure to welcome Professor Hubert Yin, our first speaker from China. Hubert received his PhD from Yale University in 2004, and then spent a postdoc period at University of Pennsylvania School of Medicine. So Hubert is the professor and deputy dean from Tsinghua University, and he's also deputy editor for Journal of Extracellular Vesicles. So prior to joining Tsinghua, Hubert is a tenured faculty member of the University of Colorado, Boulder. He's a recipient of many accolades for his research in chemical biology and drug discovery. Just a lot here. So I'm just going to read three of them. On the top, which is National Science Fund for Distinguished Young Scholars, Beijing Outstanding Young Scientists, the ACI's David W. Robertson Award for Excellence in Medicinal Chemistry, among others. So today he's going to share his talk on chemical biology investigations of EV in innate immunity.
It's a huge pleasure really to talk to you on May 21st, 2020. This is a special day, really. So in China, especially yesterday, May 20th, sounds, if I say this in Chinese, it would be 520. It sounds like I love you or 我爱你. Or May 21st, it sounds similar. So May 20, May 21st, almost like another Valentine's Day in China. So it's such a, it's a great time to talk to you about WebEV. Carolina and I were really in the same class. Right? Carolina, I believe we started serving on the ISEV board starting 2016, right? So back then I was a representative from the America chapter, but after, during this period, I moved to China. So today, what I'm going to talk to you actually is what I presented 2 months ago at one of the ISEV workshop in Argentina. I was like now, when I think back, I think it's rather crazy this year. I was traveling to Argentina in March. Really, yeah, that's really right after the COVID-19 started coming down in China, but it hasn't really starts up in South America at least. So that's really became a escape. But that's I think that's a really pretty good workshop. It's focused on, so ISEV, by the way, for you guys who are not familiar with that, ISEV organized usually at least 3 workshops a year, for different geographic locations. And this one is on immunology. So my talk then was a chemical biology investigation of EVs in innate immunity. So I would call myself, by the way, let me turn on the laser. I would call myself a chemical biologist. What does that mean? So my training background is chemistry. So biochemistry, people say it's more leaning towards biology. If you swap the word, whether chemical biology is more about chemistry. So we use chemistry too to understand important biological problems, such as these different immunology problems. And we're also interested in membrane proteins in general. Those, including, for example, some of this like innate immune receptors, such as toll-like receptors, which I'm going to elaborate. We also developed tools to dissect the transmembrane protein, transmembrane domain protein-protein interactions. Basically, we designed short peptide, goes into the membrane bilayers, disrupting protein-protein interactions. And with that too, we can understand their structure and function. So as Carolina mentioned, I used to be a faculty member at the University of Colorado, Boulder. I still remember the very first class I developed in Boulder was a course called Chemical Biology and Drug Design. So I usually like to start my introduction of chemical biology. If you're not familiar with this concept, is we're providing tools. So with that, we can perturb the biology system. It's more like if you see a student sitting on the back of the classroom, you want to tell if he or she is sleeping, you want to poke him or her. That's a perturbing. With things such as membrane protein, for example, they're hard to perturb. And as chemical biologists, we provide such tools to understand their biology, apparently including EVs. And I'm going to talk about some of these curved membrane tools or curved membrane sensors that apparently is a characteristic for extracellular vesicles. So I'm going to start actually today with the toll-like receptors. Toll-like receptors, interesting name. When I first started working in this area, I was thinking this group of proteins have a interesting name, seems from the toll booth from the highway. So apparently this innate immune receptors have nothing to do with toll booths. Toll-like receptor, this word toll is from German because in the 80s, the toll gene in fruit fly was first discovered in a German lab. They found this gene is really quite amazing. They named this toll. Toll means like awesome, like we're amazing. So when people discovered the analog, the human analog of toll gene in fruit fly, namely 1998, Bruce Butler in Southwestern reported TLR4 is the receptor for endotoxin, or LPS, for which apparently Bruce won the Nobel Prize in 2011 in physiology and medicine. They named this important toll-like receptor families through 1 to 10. We humans have 10 different toll-like receptors. So as I mentioned, immunology apparently has a lot of current interest as evidenced by the Nobel Prize 2 years ago in immunotherapy. And there's another one, as I just mentioned, 2011 Nobel Prize was awarded to Butler, Hoffman, and Stamen for their contribution for both innate and adaptive immunity. So toll-like receptors really like a major regulators in the innate immunity field. As I mentioned, 10 different toll-like receptors, they work in similar way. They form either homo or heterodimeric complex on the surface of the membrane, such as this one, TR1, 2. And they basically serve as antenna, just like different sensors for PAMP, or pathogen-associated molecular patterns, right? Such as LPS. And then after getting into the cells, they converge and turn on inflammation, the your NF-kappa B signaling pathways, such as, for example, you got infection in your throat, you got fever, that's a part of this innate immunity actually turn on, trying to defend yourself against this alien invasion, if you will. So these toll-like receptors work in a similar way. As I mentioned, TLR4 is the first TLR reported, right? Pretty much like messed up numbering system. The first one is TLR4. Of course, it's actually the gene's name. So TLR4 sends a chemical called LPS. Lipopolysaccharide is the chemical component of the bacterial cell wall, right? So when our immune system detects there's LPS around, apparently there's, with human cells, we don't have cell walls, so we don't have LPS. When there's the LPS detected, that means there's danger, there's some alien invasion, so our immune system turned up. And other things are actually pretty much similar, like for example, TR7, 9, 8, actually, things like we talked about COVID-19, apparently SARS-2 is a single-strand RNA virus also. So those single-strand RNA by our innate, detected by our innate immune system, actually through TR7 and 8. So their ligand is single, like a virus derived single-stranded RNA. And the same thing, they turn on NF-kappa B signaling and protect ourselves. Of course, all of this innate immunity, you could call them double-edged sword, because excessive activation could cause different pathological disorders also, such as different autoimmune disease. So just yesterday, we actually like one of our review paper just came online, show on this accounts of chemical research, a journal from the American Chemical Society. The idea is the same. Basically, it's like the watchman standing on the top of the Great Wall, watching this PAMP or DAMP, pathogen-associated molecular patterns, or danger-associated molecular patterns. And then when there is this danger signal, they would actually like send signals and turn on self-defense. So our lab, like 13 years ago, when I first started my independent career in Boulder, Colorado, we started focusing on innate immunity. So in the last 13 years, I can't believe that 13 years already passed. So we developed different small molecule probe. Remember, poking that guy, there's chemical tools with which we can study biology. We reported the first small molecule inhibitors for TR1, 2, TR5, TLR3, and TR8. We also reported the first inhibitor for activators, TR1, 2, as well as some other regulators for 4 and 7, actually working on that also. So for the sake of time, I'm not going to bore you to going through all of these. We wrote some reviews also. Besides that one, featuring Great Wall, there's some other reviews. So if any of you are interested in this area, please check them out. I'll just give you one example to show you what we chemists can do to help you, to probe the biological system, understand other things, like including EV. I'll just give you one example. That's our studies on TR8. So talking about toll-like, 10 different toll-like receptors. Toll-like receptors sitting on the cell surface. Basically, they sense different bacteria and virus-related membrane component. And their other group actually in the endosome. So they're called endosomal toll-like receptors, including TR3, 7, 8, and 9. And they sense different nucleic acid, basically, single-stranded RNA, double-stranded RNA, bacteria-related CpG DNA, and so on. So there have been some chemical small molecule tools for the surface TLR, but for endosomal toll-like receptor inhibitors, you basically can count them with with one hand. They're very few. So talking about the reason, of course, if you use the small molecules and probe a toll-like receptor in the endosome, those actually usually their ectodomain were found in the lumen, basically pointing inward to the endosome. They have to go through the plasma membrane, endosomal membranes, and reach the ectodomain. That's hard. On the other hand, we also think that's due to the lack of mechanistic understanding of these toll-like receptors, how this work. For example, I just stole this figure from a pretty influential review paper from Nature Review Immunology back in 2006. So the previous understanding of how this toll-like receptor works, basically, there is a foreign nucleic acid, as a PAMP, right? They can induce TLR9, this is one of the endosomal toll-like receptors, form a homodimer. So only in the presence of the ligand, TLR can form this dimeric complex, which is active in this signaling pathway. So all of the previous work trying to develop its inhibitors actually basically focus on this active form of this ligand bond TLR dimer. until 2013, a group in University of Tokyo, actually like this, our collaborator, Shimizu's lab at Tokyo University, I think they're in the graduate school of pharmacology, pharmaceutical sciences also. So they reported a unliganded dimeric form of TR8. So TR8 is known to sense a single-strand RNA. Basically, the ligand is the degradation product of the related single-strand RNA. There's two binding sites. The first site is here. Uridine binds to two copies of TR8. And then there's another site, nucleotide actually binds to those. So, but interestingly, Shimizu's lab reported, along with this agonist bond form, so-called activated TR8 dimer, there is a ligand-free dimeric complex also. So people don't actually know that, didn't really know before TLR could form dimeric complex without any ligand. So is this true? Could this dimeric complex really form in the absence of any ligands, or this is a crystallographic artifact? But that is the question. So that's how we started also. So maybe you can actually hear the background also. So this is the so-called pharmaceutical center of pharmaceutical technology, which I directed at Tsinghua University also. So we built this high throughput small molecule screening, basically screening these like HEK cells, which has pretty minimal expression level for toll-like receptors, and we knock in different human TLRs expressing each individual toll-like receptors. Because some of the toll-like receptors, such as 7 and 8, they could interfere with each other. And with these, we can have a robust assay to identify their inhibitor. So to make a long story short, we did several rounds of screening. I'll skip the chemistry part, which is boring. And we finally came up with a first generation, we named this a CuCpt8M, that is TR8 small molecule inhibitors first reported in literature with about 70 nanomolar IC50 in the cell assay. And using ITC, we showed it binds to the active domain of TR8, and it's pretty selective. Remember, all of the toll-like receptors, they look similar. They're sitting on the cell surface, basically this shape, M shape. And this one only block TR8 activation without touching other structurally similar toll-like receptors. But what's more exciting or intriguing maybe is what we can learn with these tools. So again, the question is whether toll-like receptor could actually form dimeric complex without any ligands. So we found these compounds binds to a pocket which only appear when these two copies of TR8 could form a dimer, right? So that means, so this actually is the antagonist binding site here, which is right adjacent to the agonist binding site, site 1, but it's different. So that means indeed these toll-like receptors form a dimer first, and then so that actually can be recognized. On the other hand, this is also like based on sheer luck, really, because we didn't really know there's a pocket existing there. But with these two, we confirmed indeed toll-like receptor 8 could form a dimer without any ligand, and this actually gives you the resting states. And that provides a new strategy also. So if the previous, and most of them failed, work trying to block toll-like receptor 8 activation, if you can say that is like get an open door close, we really like by accident found a key, if you will, really lock this door. So this TR8 won't get activated anymore. So this actually provides a general concept for the TR8 mechanism, and also as a chemical biology. So with that too, we actually like use this for different immune diseases, but I mean, I'm sure like many people already like think like this, how this actually related to EV. To have this very long background introduction short, we use that for different diseases, such as RA and some orphan diseases. And what we can do, if any of you guys actually interested in any of this too, so we can actually like do structure-based drug design. For example, this student Jane Yi Hu, like looking at the complex structure, it's a crystal structure. The binding site still have space here. So what we're trying to do, we install different functionality, trying to, for example, a hydroxy group here, trying to talk with the serine side chain. With this additional interaction, we improve the IC50 of this compound to lower this to like picomolar level. So this is actually really the most potent compounds we have ever developed in the last 13 years, as we said, working in these areas. So this provides a unique and hopefully helpful tools with which we can study different toll-like receptor biology. For example, one of the examples is a paper we published 2 months ago with Norwegian University of Science and Technology in Norway, apparently, the country next to Jens' country, Sweden. Basically, we showed the HIV pathology has an interesting relationship with toll-like receptor 8 with the help from this toll-like receptor 8 inhibitors. So again, that's indeed a very long introduction to chemical biology as a general rule, but really what we can do with the EVs. And that's really like coming from our interest for providing useful tools for this, some of this complex system. And this particular one is when we look at the curved membranes, right? So as I mentioned, we're really interested in membrane protein nucleic acid and the protein lipid interaction in general. And so this is what I borrowed from this classic cell biology textbook. So the conventional view would have this, for cell membrane, you have this phospholipid bilayers, you have membrane proteins floating inside, and this is the flat membrane. So we do know a lot of this membranes now they're curved. So for example, if you like thinking about this point here, if you think about to fit a sphere or like a tiny snooker ball, for example, here, and the radii or the radius of the ball you fit here actually like reflect a critical feature of this point. That is the curvature. So C1 and C2 could be defined as the reciprocal of the radii. Basically, the larger the ball you fit here, the less curvature you have. And the different combination of C1 and C2 could give you different shapes on of the membrane. So all of the weirdo shapes. Apparently, all of these shapes could be found in cells, and they do play important regulating roles also. So what do we know about this? Actually, what induced this curvature, right? This actually could be like related to the secretion and take up of EVs as well. So because when you generate or you take in some EVs, those it's almost like fission fusion process, membrane remodeling, basically. Membrane curvature is a unavoidable, if you will, problem in this EV biology. So, but talking about the machinery regulating curvature, we still don't really know much about it. So we do know, for example, some of these amphiphilic helices, for example, they can insert into one of the leaflets of the phospholipid bilayer, basically stretching the cell membrane, stabilizing a curved membrane. Or some other proteins, such as the bar domain, they can literally like grab the membrane, induce curvature. Such as the snare complex, they stabilize the curved membrane through electrostatic interactions. So one of the common tools we employ in my lab is computational simulation. We like to play computer games. A previous postdoc in my lab, RJ De Jesus. Right now, he's teaching at the University of Manila. So actually, I was supposed to visit him early this year also. But this year, 2020, besides COVID-19, there are many other exciting things, I guess, if you will. If you guys remember, especially in the Asian Pacific region, there is a volcano eruption early this year in Philippines. So I actually canceled my trip directly the day before I took off. Anyway, so when RJ was a postdoc in my lab, he developed the first curved membrane atomic like atom simulation system to simulate a spontaneously curved cell membrane. And this work was continued by a MD PhD student, Noah Castelowitz, in my lab. I joked with Noah that he was MD student. Right now, he's a resident at Stanford Medical School, but he also, in my lab, he did MD simulation also. So anyway, so with this MD simulation, Noah found when cell membrane started curving, one of the characteristic of it, it actually become like provide more defects. Basically, those lipids don't pack perfectly. They can provide some like vacuum holes on the surface also. So that is also a characteristic of cell, like highly curved membranes. Another things we want to, we're interested in is the lipid component. So I like to give this analogy that I would say for membrane biology, really like most of the previous work were focusing on membrane proteins because it's almost like to tell a story of an army. So people usually like focus on their generals and knights, like in the ancient time. But really, when the people who really went to the battlefield to fight, the soldiers. So cell membranes are more or less the same. Those membrane proteins of those generals were their knights, but really like the lipids, which we kind of like actually like overlooked for a long time as those soldiers. Human has like more than 1,000 different kind of lipids, and their distribution, their abundance, actually all play important roles, such as PS, for example. This is a negatively charged phospholipid. And in normal cells, PS were 99% of the PS were pointing inward, right? So only when cells underwent, such as apoptosis, PS got flip out, and exposing PS became so-called eat me signal, and our macrophage could actually like find them and clear them out. This is part of our cell protections also. So there are two things we know for highly curved membranes. They have enriched PS, like more charged lipids, and also more defects, as I just showed you. So, so this is one of the biological function. For example, PS exposing, this could be actually like recognized by the neighboring macrophage, as actually I showed here. This is the GFP labeled PS and the neighboring cells. The movie didn't really play. I don't know why, sorry about that. But anyway, it clears this dying cells. So based on these two features, we designed our probe, again, chemical tools. This is from the effector domain of the MARCKS protein. MARCKS protein effector domain binds to the PS enriched inner leaflet in the cells. And we think this would be a reasonable starting point for highly curved PS enriched membranes, such as actually what we could find in many EV cells, so because of the following reasons. One, this is a relatively short effector domain, 25 amino acid residues. And importantly, this domain is not structured. So you don't really worry about truncated this region and lose the secondary structure. So this region is unstructured, but upon binding to the membrane, it took a it actually take a bolt shape, actually like our group and David Cafisto's lab in University of Virginia reported the same thing, observation. Basically, there are 5 phenylalanine residues perpendicularly insert into the membrane, and then through electrostatic interaction, you can see a lot of lysines here with a positive charge. They talk with with the PS, for example, like with a negative charge. So the electrostatic interaction is the driving force. And we think also this phenols, the benzyl side chains in the phenylalanines could fill in these membrane defects as a characteristic of highly curved cell membranes and stabilizing this curved membrane regions. Also, just like what my previous students actually demonstrated on this scaffold. So using that too, so we is like a postdoc, Janelle Saluz, who is in Philippines also. Actually, he's the vice president of his university now. And Leslie Morton prepared this MARCKS-ED probe. So this is from the effector domain, and we prepared 2 mutants as control, with which we basically replacing the phenylalanine or lysines with alanine. And we show that this MARCKS-ED did actually simultaneously sensing curvature and lipid component at the same time. So this may actually provide a useful tool. And then I moved to Tsinghua. So one of my first graduate students at Tsinghua, Lei Yan, he did carried out further computational simulations also. So if our hypothesis holds true, which is the secondary structure of the effector domain is not important. You imagine OD, so like all of our amino acids, natural amino acids, they have L amino acids, right? All of the peptides actually have L amino acids. Actually, with like a couple of exceptions. But D, which is the mirror image for this, just like the relationship between our 2 hands, the Carroll image should have the same function also. So we did MD simulation and found the D MARCKS-ED also behaves similarly, basically like have the similar profile with the L marks also, lending some support to our rational design. And we also validated this in vivo. So this is a collaboration with Ding Shui's lab at Tsinghua in C. elegans. Basically, we inject fluorescently labeled MARCKS-ED probe into the gonad, which is the reproduction organ of the worm. Because if you remember, in normal cells, 99% of the PS pointing inward, so there is no staining. And we knock out a gene called TAT1. TAT1's function is to flip PS inward. So if you knock out this gene, PS remaining exposing on the surface. So to these, MARCKS-ED actually have pretty significant staining. If you look closely, there's a little bit of staining with the mutant also, so this is probably due to non-specific interaction, but compared to this is weaker. We also knock out another gene called SET7. SET7 is involving this in this eat me signal. So in this bright field of the worms, you can see this dead cell still like sitting around. So to those worms, even in the presence of these normal cells, you can use our MARCKS-ED, you can selectively label those PX exposing dead cells in this worm. So showing this could work in living system. All right, then finally, which is like I believe this is the first slide of EV I showed today. So so EV, of course, like we like there are many experts sitting in this Zoom room, I believe. So this actually like doing important roles in cell-cell communication. For example, Yan's previous beautiful work showing EVs carrying out these RNAs in the circulation system, triggering on different biological response. So and also the EVs in in cancer development, for example, they direct metastatic cells to remote sites, such as this 2015 David Layden's paper, for example, showing the integrin expressing on the surface of EV, directing those EVs, actually like moving the tumor cells to either lung, from breast to either lung or liver. So our when we first, this is about 10 years ago, And this is technology like basically we used with our chemical probe. So we're still continue working on this technology with my collaborator, Fred Lee, who actually used to be in Australia, also studied at University of Melbourne. Fred also worked with Andrew Hill on this very beautiful approach on Alzheimer early detection. And his startup is very successful. So we have another venue in Shanghai, based in Shanghai also, focusing on cancer detection. So our hope is really to collect some body fluid, right? Because all of the EVs were found in body fluid. You guys know about that: blood, urine, saliva, breast milk, and so on. So one of the feature apparently for especially this, I would say this is not only a diagnostic application, maybe some prognostic also. Basically, we all know that breast cancer became has already become like a chronic disease in some sense. So really sometimes not only diagnosed, like whether a patient got cancer, but also whether this cancer has potential to move to metastatic stage. So talking about this, I still remember this article I read in Wall Street Journal a few years ago. They're talking about, I think it's shocking, only 20% of the patients received beneficial treatment after they were diagnosed breast cancer, saying actually a lot of this actually were really unnecessary, like doing more harm than help to the patients. So our hope is really to collect some of this body fluid and adding fluorescently labeled MARCKS-ED, for example, upon binding to the highly curved PS-enriched surface of the EV, we can have a very quick, cheap, non-invasive technology to determine their EV levels. So these movies didn't really show, but anyway, we test this in pathological animal ex vivo systems. We showed in the scattering mode, there are many different particles, and when we switch this to the fluorescent mode, there is no fluorescence. And to the same sample, we added our probe. Again, this is the MARCKS-ED with fluorescently labeled. So it's still the same sample, but only a subpopulation with a small sized. So talk about like the exosome. If we talk about exosome, 30 to 100 nanometers particles were selectively stained. So this one, again, we continue developing this in Shanghai. And there is the further, this story didn't stop here. So last year, a previous student, who Tamura, who is actually at Sloan Kettering now, like trying to use this as delivery tool also. So we attach this MARCKS-ED as EV finding particles conjugate with a warhead, if you will. It's doxorubicin derivative, which is highly toxic, as you know, but they might be too toxic because they kill normal cells also. So we link this with a light-sensitive linker. Upon shedding UV light, it would release these toxic agents when they find these cancer cells, which shed a lot of EVs. And without UV, they're actually basically intoxic. So we showed this indeed find EVs, and also they get into cells. Both L and D, if you remember, have two versions of this MARCKS-ED peptides. With EV and without EV, the toxicity of the agents increased about 1,000 folds. And as a control compounds, peptides that doesn't have this selectivity. Carolina told me I had about 40 minutes, so I would just use like last a couple of minutes to finish a recent story. This is coming back to toll-like receptors. So please don't blame me to waste too much time on toll-like receptor at the beginning of this talk, because I meant to actually come back here to innate immune system. So remember, I talk about like when we first talk about like starting of this this talk, we talk about LPS activated toll-like receptor 4, right? That is the surface immune receptors. But in 2014, there is actually a series of very interesting stories from, it's called the National Institute of Biological Science in Beijing, China. Feng Shao's lab, they showed LPS has another non-canonical pathway. Besides the surface activation, they could get into the cells also, bind to the caspase, and turn on pyroptosis, which is similar to apoptosis in some way. And also, interestingly, in this in this paper, 2016 Cell paper, they also showed outer membrane vesicles, which I guess you can argue that is the kind of EVs wrap up those LPS getting into the cells. So could be one of the things like this PAMP or pathogen-associated molecular patterns depends on different particles getting into the cells. And we're asking, is Yin Zhang and Hubert Yin in my lab asking this question, is that a general phenomenon? Those PAMPs depends on EVs to get into the cells, activating innate immunity. So we use this Cre-Loxp system, which I believe many of you guys are pretty familiar with. We're trying to look at the EV transfer so if the EV donor cells actually release EVs, if they were taken up by the recipient cells, they would actually removing the roadblock, and the GFP would express, and when you see the color change, that shows EV successfully transferred. So Yin and Hubert showed that either you mix those donor and recipient cells, or you actually have this transwell, basically have a distance, separate them to mimicking either in proximity or in some longer distance. Indeed, actually it's ODN, which is the ligand for TLR9. The native ligand for TLR9 is the bacteria-derived DNA. Seems depends on EV, and they could act when you activate the cells with ODN, they release EVs containing ODN, and later on accepted by the recipient cells also. And then we also test those EVs in the controlled, the EVs from the controlled cells and the ODN activated cells. And using this proteomic studies, we found a few knot, if you will. One of the central pieces is this thing called CDC42. CDC42 is a cytoskeleton protein. Again, when we talk about the membrane remodeling, this could be important protein. So it seems CDC42 play important roles in this process. And CDC42 proteins and also mRNA were found in those activated EVs also. So we showed CDC42 is important if you use siRNA to knock it out. It actually reduced the cell transferring, and we also confirmed this with this EV small molecule inhibitor GW4869. However, actually also mentioned, since this is the talk discussion also, we also have concern about specificity of GW4869. It seems this is a pretty non-specific inhibitors of the EVs. Anyway, also looking at the construct with the constitutively activated CDC42 and showing indeed CDC42 is involved in the taking up and also of the transferring EVs. Another thing we look at is this philopodia. Basically, it's a membrane, the remodeling on the surface. When you activate the cells with ODN, you can see the numbers of those, the bulging basically increased, and this could be blocked by QNZ, which is the TNF alpha inhibitors. So to make this long story short, I'll just actually sum up here with this studies. Actually, we already published this last year. Basically, we think other toll-like receptors such as TLR9 ligand, just like LPS, get into cells using OMV. ODN may actually also transferring with the help of EVs, and also after taken up by the recipient cells, the CDC42 proteins also has a feedback and also promote further uptake of EVs. So this, this could actually like suggest maybe some of this PAMP, the different ligand for toll-like receptors have a rather common feature, basically taking advantage of the EVs. So I usually have a couple of slides to advocate ISEV and JAV, but since Carolina and Yin are here, Yin is my boss for Journal of Extracellular Vesicles. I'm one of the deputy editors. I would skip, and also I think I'm over time already, I would skip those slides. But also, since Carolina mentioned I'm the first speaker from China at this series, we do have a Chinese society also. So last year we had, I just trust all of you guys can read Chinese apparently. So last year we had our third annual meeting in Xi'an, China. We have like about 500 something participants in Xi'an. And one more time, thank you very much. I'm sorry to going a few minutes over time. Yeah, back to you.
Thank you so much, Hubert.
Thank you.
So this was really interesting, and digging into the details and the ultrastructure of EVs is really important. Yes.
Okay, this is fascinating work, especially that I'm not from that field at all, so it's like a space science fiction to me. But generally, does your molecules that sense curvature of the membrane, are they specific on the angle of the curvature? So are they equally efficient in sensing curvature of small vesicles and larger vesicles?
So when we talk about curvature, first of all, I should actually start it. Curvature sensing is a pretty general phenomenon in biology, right? Such as the snare complex, as we know, they like have the C2B domains. They have different function of. Sorry, my boy was background. I all have the same problem apparently these days, and like I was told that school will remain closed. So so for our design, this effector domain from the MARCKS, they sense the optimal one is around 100 nanometer diameters. So our ultimate goal is really to design any like a probe for any given curvature. Basically, like for example, you're interested in exosome 30 to 60, let's say just randomly to call a number, or 60 to 100, because the size, we do know, we do think the size of EVs matter importantly. Like they have the important biological roles. So we hope to develop computational method, like some kind of AI system, if you will, to design probe specifically for any given size or chemical component. Of course, that is the long-term goal. When we come to lipids, the molecular recognition is lagging much behind compared to protein and nucleic acid in general, because actually like lipids, protein-lipids interaction are more difficult to simulate in general. So right now we do have this probes favoring smaller particles, basically. That is, in other words, that's high curvature. And also they usually prefer membrane with more charge, like PS in particular. But there
okay,
are Go ahead.
Yes, sorry. One more question. So that MARCKS actually cannot bind on the part of the membrane that is highly packed with proteins. So as more proteins does the membrane have, the less binding of the MARCKS would be. Am I right? Since it's only getting in the lipids, right? Proteins hinder the binding, yeah?
You're talking about the peptide probe or the protein itself, the MARCKS protein.
Yeah, but if other proteins are already in the membrane, it cannot get into the lipids, right?
Yeah, that's a good question. I don't know, because if we talk about EV, they're really so the size of the peptide, if you act like we did like the simulation, if we're looking like really like the smallest EVs, the size actually is comparable. So if you think about this particularly, so I would say definitely there would be some competition, but the binding affinity of lipids with protein is a more tricky issue. People usually use the ratio, basically like how many lipids per protein, because when you talk about, if you're micromolar, this kind of bulk concentration, it doesn't really actually it's really relevant, because it's only like on the cell membrane. So that's one of the features for membrane proteins.
Yeah. Okay, thank you.
Thank you. So I have a general question I was about to ask in the beginning. So when you think about the curvature, you think the lipids or the proteins in the membranes are the most important for maintaining that ultrastructure of the vesicles?
Good question. I don't know. This in cell membranes, definitely the different machinery regulates this, right? One of the perfect example is the Jim Rossman's snare complex, and
we can open vesicles, for example, and then we close them again with adding energy. So we open them, keep them open, and we close them again, and they maintain their topology. They don't flip-flop or anything like that. So that argues to me indirectly, I guess, that proteins might be important for that topology.
But talking about this, both positive curvature, like those bulging ones, and the negative curvatures, there's specific protein machinery regulating this different process. So I would guess for EVs, if we're talking about, especially in their generation, both cases could be relevant, right? I don't really know. There, of course, different people propose different machinery. I don't really know. There are a lot of general understandings for different positive, for example, positive concave or negative curvatures were regulated by different machinery.
Okay.
Yeah.
So I have a few questions, and I'll come back to, but I'll begin with Faiz Shikari. So please unmute yourself. Maybe I can do that. I do unmute you, and you can speak.
Okay, thanks for the nice presentation. I have a naive question. You showed that phosphatidylserine can affect the curvature of EVs. On the other hand, we know that some kind of methods, such as ultracentrifugation, can change the phosphatidylserine localization into outer membrane. So is it possible to have different EVs shape by different methods of EV isolation?
That's a very good question, Hubert. So if you think about it, when we use a fixed angle rotor, the vesicles will be in the tube and they hit the wall, right? And they start rolling down or slipping down that plastic. Does it change the lipid structure, or is that responsible for some of the flip-flop topology that we see? That's basically the question, right?
Good question. Yeah, I'm thinking. Actually, when you asked that, I was thinking hard whether we know anything. Different method give you meaning,
I don't think we know that. I don't think we know that. So one way of testing that would be to have that fixed angle rotor, do your centrifugation in that, and then have a swinging bucket rotor with a cushion in the bottom, so they don't impact into the plastic. So they have a softer, and potentially that could test that or answer that question.
Good point. I don't know, really.
And maybe size exclusion chromatography.
Yeah, exactly. So that is not less impactful and less G-force, right?
Another thing, but I mean, actually, this is not answered by any chance. But I would just one comment maybe. I was thinking sometimes we just presume that all of the EVs more or less spherical, right? They're ball shape, sphere shape.
They're not.
They're not indeed. We actually see for example using EM different shapes, like donuts sometimes. So That's we don't know.
Yeah, I mean, if you do cryo-EM, most of them are round, most of them are single membrane, some of them are double membrane, some of them are small oval, some of them are extended with actin filaments inside, and they're just, you know, very diverse from that perspective. So, and that doesn't seem to change with the isolation method, but it, you know, maybe it should be tested further. We'll move on.
Yeah, sorry.
Yeah. And I'll mute you. And then we go to Bernardo Villarreal Ramos.
Yes, that's me. Thank you very much for our presentation. It was very interesting. I'm pretty much getting very interested in this field. And hence the question of different cells producing different EVs. And the process that I'm thinking of is cross-priming in dendritic cells and also, for instance, epithelial cells. To what extent do immune cells differ in the production of EVs compared to, say, fibroblast or any other cell?
I don't know. Yeah, sorry, like I don't know, but I do know.
Clearly, vesicle cells from different origin produce fundamentally different EVs with fundamentally different molecules. So a mast cell will produce very different vesicles from a HEC293 cell or a dendritic cell or a epithelial cell. So even the same cell can produce multiple subpopulations of vesicles, and those subpopulations may be different in different cell types, and this is really opening the big question for future research, I think, where we need to try to understand that difference between different cells and their subpopulations there and the structures.
But yeah, like I meant to say that, however, even the same cell type, like under different conditions, their shape could be different. Because today I meant to actually talk about shape. They could actually undergo drastic shape change, and you know they're like coming to their EVs. There might be like, you know, it's conceivable they provide different EVs also.
I guess that it comes back to what Jan was talking about. The constitution of the EV will pretty much define, and different processes will induce different ratios of protein versus lipids in the EV.
Even when we look at exosomes and microvesicles and the different ways to isolate them, we see fundamentally different cargoes. So exosomes are not correlating directly with the cargo of the donor cells, whereas the microvesicles do, if you look at RNA, for example. So it's an amazing topic, and it's so much work that needs to be done there. I think we'll move on to Genevieve, who has two questions, I believe. You need to unmute yourself. I don't have you on my screen. Okay. There you are.
Sorry. Actually, probably I'm not very well awake. This is extremely interesting. It just hit me. So what exactly is CDC42? How is it involved in cargo uptake? Because I kind of assimilate it with cell division. So is it acting on the cell surface, or is it once it's the EVs have to be taken up first?
So CDC42, we actually at this moment, we don't really know whether this is a cargo specific, if you will, mechanism. So we do know that TR9, like the TR9 like an ODN transferring seems depends on CDC42. This was actually kind of supported by a previous study by a group in Cornell, 2016 also. The EV production seems like the CDC42, by the way, is the enzyme also. That is actually, like, again, as I mentioned, actually involved in the membrane remodeling. So we think this cytoskeleton related enzyme is related to at least some of this PAMP, like toll-like receptor ligands, transferring from one cell to the other. But whether this actually apply to other cargoes, you know, for example, other PAMP or in general other cargoes, I don't know. Like this, we only started this. We only know this actually seems actually found in the TR9 activation process. If this answers your question, I'm sure. Yeah.
So you mean this is kind of immune cell specific? It would not happen with entry into other type of cells. for instance, epithelial cells or?
We use macrophage. I don't really know what about other cells. Good question.
Okay.
I don't know.
Thank you. I will look into it. My other question is kind of very naive. I was wondering if your MARCKS system to detect EVs, if you have developed it into a capture system, or if it can be done.
Yeah, you have our business idea. I was working with Fred on that right now.
Mm.
Actually, right
Okay.
after this, I'm flying to Shanghai to discuss with him about this, its potential.
Thank you.
Okay, so I'll be looking into it. Thank you.
So we will shift to Lin Li. You've asked questions before. I know we can hear you if you unmute yourself. If Lin Li is not there, what's your comment about the appearance of PS on different types of EVs from different cells? Are you there? Yeah, now you're there.
Yes, wonderful talk, Professor Yin. Yes, could you please give us some comment about the presentation of PS on different type of extracellular vesicle, like microvesicle or exons, or like the appearance or the amount of PS? Maybe it depends on different type of cells. I'm not sure. Thank you.
So you know that like ISEV, like for different workshop or annual meeting, we have an education day, right, program. So in like for a couple of times so far, I actually like at education days, I talk about the lipids, like in EVs. But I guess if you haven't been any of these, you didn't really like miss too much, because to put that short, we think like as many other things in EVs, we think many factors could actually like change different things, including the preparation method, storage method and things. Especially when we come to lipids, this is really difficult. I would actually recommend this review article, 2018, from Alicia, how do you say her?
Alicia Weaver.
No, not Alicia Weaver.
Alicia Lorente, I think, from Spain.
Lorente from Norway. That's right.
That's
right.
Norway.
Yeah, she has, but she's a Spaniard herself. She published a different, pretty good lipidomics analysis for different cell, different pathology conditions, and their lipid distribution in EVs. I would recommend that as a pretty good reference. But really, like again, as I said, varying factors. That's so common in all of the EV research I found.
If you send us that link, we'll put that link into the YouTube YouTube channel, so people. We shift to Nader Kameli, who has a question about the Transwell system. Please unmute yourself. If not, I'll read the question. How big are the pores in the Transwell system you use, and how specific is it, I guess, for EVs and other molecules?
Yeah. I don't know.
Have the
maybe in the first author of that paper, I can't recall. Are you online? I think she was talking about she was coming to join in. Are you maybe?
Ying Zhang is there.
Oh, do you know him?
Yes, I know. It's 400 nanometers.
Okay.
Nanometers.
So how does it influence? What vesicles do we get, and what other molecules pass over? And it's a beautiful system to prove something.
Mm-hmm. So it's selective for smaller vesicles. So because we use the diameter for the Transwell, it's 400 nanometers. So only the size of vesicles smaller than this could pass the Transwell. I think, yeah, that's it. Okay.
Thank you.
Thank you.
Mike O'Neill, he's very active in trying to understand what's going on in EV field and in the COVID field. Can you unmute yourself, Mike? Mike is basically writing a lot of newsletters and things like that. So we've interacted with Mike for many years now through ISEV, and he's attended my ISEV meetings multiple times, and now he's, presenting a lot of new research. Now we hear you, Mike. Go ahead, ask a question about.
Can you hear me? Can you hear me all right?
Yeah,
yes.
I can hear you, Mike.
No, I was wondering if TLR8 interacts with the COVID RNA, and also how TLR8 might, how does it distinguish between viral and host RNA?
Remember, I mentioned this paper we work with this Norwegian group early this year on HIV. So like after that, this is called Truth Flow, like her group Truth Flow. We started like, working on COVID, like SARS-2 also. Apparently, like I think it's got to be like single, because SARS-CoV-2 is a SARS-2 is a single-strand RNA also.
Right.
So yeah, TLR8 is the machinery sensing virus single-strand RNA.
So I've got to extend that question and ask whether, so we know that this virus can, in some instances, induce a cytokine storm, right? Which potentially could be mediated by the TLRs. I presume TLRs are important for that. Mm-hmm. Are there differences in the structure and the efficacy of different viral RNAs to activate the TLR4? And could that explain the extremely high potency of the SARS-CoV-2 virus to induce cytokine storm and deaths in some cases, Hubert?
I haven't really
difficult questions, I know.
Yeah, we right now I people have already like 26,000 papers published this year on COVID-19 related topics, but I haven't really
26,000. 26,000.
Talk about this like a tsunami, like
yeah, it's a tsunami of papers. I know. It's impossible to read, right? Yeah.
No way. A lot of this actually like But anyway, so I haven't really read a lot of this innate immune, with molecular mechanism. To be honest, I have to say some papers actually, like I don't say like all of them, but some papers are actually pretty low quality. I don't really trust a lot of this data. But TLR8 and 7, these are two actually like related. In HIV and other viruses, they definitely like play important roles, and also they're known to induce cytokine storms also. That's for sure. I don't think COVID like SARS-2 is
probably mechanism, right? Yeah, okay, that concludes the general questions. Thank you so much, Hubert. Carolina, some closing words, please.
Okay, thank you so much, Jan and Hubert. Wonderful talks, and it's lovely to see you here. So next week we have our first speaker from Japan, and he's from Dr. Yu Fujita from Ochiya Lab, and he's going to talk about lung fibrosis and how that relates to COVID-19 as well. So I hope everyone come and join us again next week, and stay safe. See you then.